Welcome to TechBio Talks, where we explore the bold ideas, breakthrough technologies, and visionary people reshaping the future of AI and medicine. My guest today is both a pioneer in the field of image-based profiling and a fierce advocate for open science, a combination that has led to hundreds of labs across the industry adopting her team's tools and data sets to accelerate drug discovery. She's also one of our earliest supporters and scientific advisors at Recursion. In fact, her work is so closely tied to the founding of our company, and we'll get into more of that later, that we named one of our labs in Salt Lake City in honor of her, the Anne Carpenter building. Currently, she's an institute scientist at the Broad Institute of MIT and Harvard. I am so excited to have this conversation, so let's get into it. Welcome, Anne Carpenter.
So excited to have a chance to catch up with you, Chris.
Likewise. So I remember back in October of 2013, my co-founder Blake and I were at some sort of drug discovery meeting in Cambridge, and we saw you, and you were like the most exciting scientist that we had ever wanted to meet in the field. You had pioneered this new kind of omics called phenomics. And we, as I recall, tried to get you to go to dinner with us to talk about science. And you said no. But let's start there. Maybe as a good place to start.
Oh, great. Let's start with the most embarrassing story. So I was a very young professor at the time trying to establish my lab. And that was the first meeting that I had brought a postdoc with me. And my one goal for this entire conference was like, make sure my postdoc meets very important people and, it has the opportunity
Which was
to network.
Not Blake and me, not Blake and me.
It turns out a couple of grad students who hadn't even finished their PhDs was not high on my agenda. So I was like, oh, sorry, we have plans, because we did have plans to like find somebody better to go to dinner with, right? So it's embarrassing in retrospect, but turns out everybody who goes to Boston to a conference like knows somebody, and I was a young professor. I was not a VIP to the others. So a long story short, it was like 5 o'clock and we hadn't found anybody. I was like, oh, maybe I can find those grad students because they seem cool. Like let's go see if we can find them. And like, thank goodness we did, because you guys told us that was before Recursion existed, maybe even before you had a name. And so wow, yeah. In retrospect, that was an excellent choice, and really glad it happened.
Yeah, we actually came up with the name for the company when Blake and I went out for drinks after we had dinner with you. But it was an amazing dinner. We learned so much about the way you were thinking about the field. It felt a little bit like a dissertation defense. You asked us a lot of questions, which was super fun. As you think back to the time when you were pioneering this field, like what initially led you to believe in the incredible potential of images of human cells as this breakthrough path to understanding biology and chemistry?
Yeah, when we met, I was excited about the potential, but I would not have, it was not certain. It was definitely an uncertain time. And so, for me, I had grown up in a generation that had just discovered mRNA profiling in the late 90s. And so profiling was really a new concept, and I just was absolutely in love with the idea. So then when I got into images, I was like, hey, why can't we use the cell's morphology, which as a cell biologist, I had already fallen in love with. I knew it was incredibly informative to my eye, but like, what if that could be extracted by computational methods? And what if we could use that for profiling? But it was by no means a done deal at the time. It was clear there were some studies. The most important one was Perlman 2004 from Mitchison and Altshuler and Wu labs. And it was clear that it could work for really strong phenotypes, but it was not at all clear that it was going to generally be useful. So I spent some of my startup money, I spent some of my postdoc's time working on it. Nobody's main project because it was too risky at the time. But over time, it became clear like, this could actually be just as powerful as mRNA profiling. And yet we're using a different image type and a different data type, and therefore getting really different complementary information. So I think it wasn't really until 2016 that my postdoc showed me the moment that I knew this phenomics image-based profiling was like really going to work for real stuff in the field was when my postdoc Mohammed Roban showed me this circular dendrogram of around 100 genes that had been overexpressed, and they fell into such delightful clusters that as a biologist, I knew matched to real biology. It was like literally like a teary-eye moment that, hey, this is really going to work. But you guys were ahead of the curve because that was 2 years after you guys had launched Recursion.
And as you think back to like the beginning of your work in this space, there were many people, not many, there were a subset of people in our field who were interested in images. One of the things that you did that I think is really fascinating is that you actually built tools and open sourced them for other people. I remember in my dissertation and even the early days of Recursion, we were using Cell Profiler and Cell Profiler Analyst. Talk a little bit about what led you to make the tools themselves. And do you think that ended up fundamentally shifting the way that this technology was adopted?
Yeah, for sure. I mean, I think everybody's career makes more sense maybe in retrospect than it does as you're going along. You just make decisions as you go. But if I look back, it actually is a really nice narrative that we spent the first maybe 10 years of my lab making software that would allow people to get information out of their images. And then the next 5 years or so making data sets that we could actually tell, are we doing a good job at matching phenotypes and matching genes to compounds and genes to genes and so on. And now the past 5 years, we've really shifted to what are all the different applications we can actually do. So I feel like we spent a lot of time building the foundations of this field to even be able to do what we're doing now. But this is really the fun part for me is accomplishing real tasks in the drug discovery space.
I think you've done a lot of great work to show the field that sometimes you won't find what you want in a specific cell type or a cell type that's not associated with a disease. But very often you can find the relevant biology. And that's a huge surprise to traditionalists, I would
Yeah.
say, in
Yeah,
the field.
we find, for example, we find the vast majority of FDA-approved drugs produce a cell painting phenotype in just a regular old cancer cell line growing in a dish. That is not something that I would have guessed or expected 10 years ago, but that's the data, that's the reality. And so, like, goodness, why not pick all the low-hanging fruit first, and then we can move to the diseases that actually do require those much more complex model systems with those trade-offs that come with it.
And you've talked publicly about your purpose, which is centered around creating quality data and sharing strategies for drug discovery applications versus building the models themselves. What would you say academia's role is in pushing the industry forward? What would you say your role is in pushing the industry forward?
Yeah, it's easier for me to answer, for my own lab. I think for the whole field of academia, I think the world works best when everybody finds a cool niche and does their thing, the thing they're passionate about, the thing that really matters, and hopefully getting input from the people who can advise them on what really matters. So of course, in our space, it's really crucial for machine learning labs to be pretty connected to biology labs and drug discovery labs. But speaking for my own lab, we personally have not prioritized lately, like dorking around with model architectures and trying to figure out how to improve the training or the training of these kinds of models. It tends to give you like percentage point increases over time. But I think for me, given my unique niche in academia, the thing that we can do best and most helpful to the community is to work with drug discovery researchers to figure out what tasks are most important to solve, and then what data do we need to create a benchmark that then the machine learning people can go to town on. But really, it's like framing the problem, I think, is a real bottleneck, and that's what we've really decided to focus on. So I would say deciding what data to make is by nature determining the problems that you can actually solve well, and that's really where we like to live. And I don't think anybody disagrees that biology is data limited. I mean, we have certain large data sets, but data sets that can answer particular questions like will this drug be toxic, or what's this gene's function, those kinds of things, we still have plenty of data that could be made.
And what kinds of data do you think are most important for the field to make next? Like what is on your list for the next big data set?
Yeah, so I mean, we have lots of ideas of things we would like to work on, but I think one that is very timely, because there's been a lot of discussion about reducing the use of animal models, partly because people care about not using animals, partly because they're very expensive, and partly because they actually just don't work that well. We have so many drugs getting to human clinical trials and then failing for tox. Not as many as in past decades, to be fair, but it's still a sort of terrible problem. So that is a big goal of this data generating Oasis consortium that Recursion is actually part of, and that I'm leading together with a colleague at Bayer on the agrochemical side. So actually, this is a problem that both pharma and agrochemical really wrestles with: is how can we make sure we produce chemicals that are least harmful to human and other animal species. So unless we're trying to kill them as pesticides, of course. So it's a cool consortium because it is also multimodal. We're gathering cell painting image data, transcriptomics, and proteomics, which is usually we're not able to do at very much scale. So pretty excited to do that in some different liver cell models. And the whole goal of the consortium is which of these data types and which of these cell models can best predict actual liver outcomes in actual living organisms. So I think that'd be a huge win towards reducing the cost of drug discovery, which of course is a major goal for our industry to be able to keep making medicines.
And I know this is likely to be sort of an open source data set. You've done a lot of work in the open science space. Have you seen the world shifting here? And in particular, you work a lot with industry. Are you seeing the perspective of industry shift as it relates to open source science over the last half decade or decade?
For sure, at least in the domain I've been aware of, it has been really increasingly common that companies are excited to contribute to these consortia that make data. And I think largely it's because the scale of the data that we might be making is just out of scope for what they would want to spend the money. I mean, they could make the data set if they really wanted to, but they can't do everything they want to. So chipping in a little bit to something that is beneficial to the whole industry eventually. Usually there's some built-in exclusive data period that can sweeten the deal for them. But I just find this like a really awesome public-private partnership, just a really great way to do science, not just because the pharma companies are helping to fund the overall project and make the data and make it available to the whole community, but also because we're working directly with the scientists who care about these tasks that we're trying to accomplish and can keep the whole thing guided. It's just like way too easy to go off course when you're trying to make data or definitely analyze data if you're not connected enough to the drug discovery folks.
I have to ask, what does it feel like, going back to that October in 2013, when you went to dinner with Blake and I because you couldn't find anybody else to go to dinner with. And now here we are, a little more than a decade later, and your team is working with, or there are groups at virtually every large and medium-sized pharma company in the world that have adopted the technology that you pioneered. Like, just how does that make you feel personally? Is that really rewarding? Tell me a bit about that.
Oh my gosh. Yeah, it is just like really the most satisfying thing. I mean, clearly folks who go down the nonprofit route, like, we're not after money, we're not after fame. Like we really just want to make a difference in the world. And to see the software being used throughout academia and industry, and to see the assay having put drugs into clinical trials, like, I mean, there's really nothing like it. And I feel so grateful because I couldn't do it on my, I couldn't just make a technology and prove it and use it, and that's the end of the story, right? It's this entire community, yourself proving like, hey, this really works for phenotype discovery, and the rest of the pharma is adopting it, trying to use it for like mechanism of action determination and those kinds of things. Just incredibly satisfying, and I'm like really grateful to you and the whole scientific community, especially in the pharma industry, for giving these new technologies a try and sort of making my life work meaningful.
Well, we're all grateful to you. One of the things, you've always had a good sense of humor, and I saw recently a bit of a cheeky post on LinkedIn where you were rebranding your work to date as Virtual Cell. And as a team that is working on the race to the virtual cell, I very much appreciate the cheekiness there because, we've gone through all of these cycles of terminology and ideas and hype, et cetera. But specifically, what issue do you take with the idea of
Yeah.
virtual cell?
Well, if there's a race to the virtual cell, then I would say, like, I don't know where the finish line is. It's like more of a mosh pit at this point, and like lots of people are doing lots of cool things, but like, what is it? Is really hard to pin down. So, I mean, honestly, the buzzwords keep accelerating over time because when I started my lab, everybody was all about the big data, and everything was big data. And then just a few years back, foundation models were all the rage for like 12 months, and now it's like, oh, virtual cells. We don't call them foundation models anymore. Now they're virtual cells. So um, I think I think what I find really discouraging about how people are using the term virtual cell right now is that it's so narrowly defined,
Mhm.
that it is not generalizable necessarily to even new data sets or to new tasks, which I would think if you've got a virtual cell, that should be something that that can do a lot of, it models a lot of different things. I mean, the term originally comes from like decades ago. I'm old enough to remember the systems biologists with like lowercase systems biologists have been using that term to define the idea of like actually working out the equations of the biochemistry and the biophysics of, can we model a cell from like first principles? Can we predict cell responses based on this like fundamental understanding of those reactions that are happening within a cell?
How do you think we should evolve the definition? I mean, should we expect that success in building a virtual cell means that one can predict quite accurately how any perturbation, including out of distribution perturbations, would affect a cell? Because I think, to your point, if you had a fundamental understanding of the biochemical interactions that are happening across every single, from atom to protein, perhaps even at the pathway level, you would be able to do that. Is that success in this space?
I mean, that is definitely success. I mean, we can define some like nearer term, actually plausible things that we could do sooner than that. I think that's reasonably fair. At this point, I'd be just happy if we just broadened the definition a little bit, right? So
Okay.
right now, folks are like, I made a virtual cell, and it's like, oh, hang on, this is actually like an mRNA level prediction algorithm. Like when you put in a perturbation of a gene or a chemical, it tells you what the mRNA levels are. Okay, that's a nice thing to have, but maybe we don't need to call it quite that. So yeah, it would be able to predict morphology, proteins, mRNA, and ultimately like function. I think
Mhm.
is what we're all really getting at. That would be great. But I mean, at this point, I would just settle for something that can definitely generalize to a new data set, can definitely generalize to perturbations or genes or compounds that have not yet been directly tested or are not super similar to something that has been directly tested. Like I'll draw the finish line there so we can at least get somewhere in the next decade or so.
I love it. Well, changing gears just a little bit, the name Recursion came from a discussion Blake and I were having after dinner with you around fractals and this idea of like these small experiments that you could do many times in this self-similar way to give you some insight into the broader complexity of biology. And I think today we do lots of experiments that are part of a broader, more macro level experiment we're doing to try and understand what drug might work for each disease. But there's this meta level experiment we're doing at Recursion, which is to try and ask the question, can we build a machine to try and discover medicines at scale in a more fundamental way? That's what I am focused on now, almost, a dozen years in. What
Yeah.
is your
Well,
big macro? Yeah.
yeah, can I interrupt you there just to say that like that systematization was really pretty prescient. Like, I mean, everyone's talking about it now, like, oh, AI agents are gonna mine all the data and discover new things, and we're just gonna like start turning cranks and medicines will pop out. I think what I love that about the risk that you guys took 10 years ago is let's find a way to find medicines in a more systematic way. And it was tied to a particular technology at the time and a particular time and place of what techniques were available. But you guys actually made it work. And I'm excited now to layer on some of the more serious, like actually working kinds of technologies that are coming out, like to see how you and others apply those to the new data we have. I mean, it's a really exciting time to be a scientist, because I think most of us, especially if we've been working in drug discovery too long, you just kind of assume things are going to get like incrementally better, and we are getting towards a point where we might see a really major shift. And that's so important because, as I said, we got to get the costs down so that we can make more medicines cheaper, so that society can expand out to what is curable.
Yeah, I agree. I'm curious for you over the next decade, like what is the big bold bet? What are the key themes that you see, the big hypothesis you want to test or you want to see tested?
I have like really one big experiment that I would love to happen in the nonprofit world. And that is, I would love to see every existing drug tested for every existing rare diseases. Because for all we know, there are thousands of rare diseases. Most of them have not had a scientist dedicated, much less, hundreds of scientists dedicated to them. And for all we know, they might actually be relatively easy to cure or treat with existing drugs that we've already got available to us. So, okay, so that's a nice experiment, but like, how do you actually do it is the hard part. But there already exist collections of drugs. That part's solved. There's around 5,000 drugs that have either been approved by the FDA or have gone into human clinical trials. So we have some data about how they respond. And so now the only bottleneck really is finding a phenotype for every rare disease. And of course, Recursion has gone out at this for quite a while. There's an NIH consortium we're part of called IGVF, which is underway that is at the moment, it's on pause because the federal government isn't paying out grants at the moment in a timely fashion, but its goal is ultimately to identify phenotypes across a wide spectrum. So if you've got this mutation that we know is causing your disorder, how does that impact the abundance of the protein that the mutation is in, or protein structure, or protein localization? And since I'm involved, also, how does it affect cell morphology? And so those kinds of systematic approaches, I think, are really great for a nonprofit universe where we can find all the phenotypes. If we can put them into pools to make it like a really efficient screening format, it starts to become actually pretty financially feasible to test all the drugs times all the rare diseases.
So, like, what kind of price tag are we talking about here?
Yeah, so it's probably, it's hard to estimate since we've never done it. Society has never done this before. But I think based on some pretty decent data points, it's less than $10 to test each drug disease combination. So for each disease, if you're testing 5,000 drugs, then you're talking $50,000. Not so terrible. You got to do it in a pooled way to make it like financially efficient. So if there's thousands of diseases, then you're talking on the order of maybe three or $400 million to do all the testing. You got to find the phenotypes first. I mentioned the consortium that's working on some subset of that, but we need to scale that part up too. So I think if you put that all together, probably $1 billion, which is like insane for an academic grant. And even most philanthropic gifts do not quite hit the $1 billion mark. But depending on the hit rate, it could be like a tremendous bargain if we can find hundreds of treatments for disease in just one giant experiment here. $1 billion is how much it costs to get a drug on the market, like a new drug on the market. I feel like as a society, we could spare $1 billion to give something like this a try. So we've launched a consortium called Vista to run like a really small pilot experiment and just at least start getting a sense of what the hit rate could be to hopefully motivate scaling up something like that.
I love it. Well, I hope that moves forward. And to the extent that it does or doesn't, I hope you know that we are doing a little bit of that work in the background. Before we go, I do want to give you an opportunity to share a free plug on anything you're working on that we should all keep an eye out for in the coming months, work by others you want to highlight, just anything that's like on your radar that you want everybody to know about.
Yeah, I would say if you are interested in data sciences and you're tired of mRNA profiles, maybe you should check out image-based data because it's an uncrowded field and where you can make a ton of difference. So a couple of conferences you might want to check out are Cytodata, which focuses really on the data side, and then SBI2, Society for Biomolecular Imaging and Informatics. Both of those are really aimed to see how can we get the most out of images to accelerate drug discovery.
Well, Anne, that was fantastic. I know we both have hundreds of millions more experiments to go run. So with that, we'll probably end this talk. But I really enjoyed hearing in particular about your purpose. I have a sense of what your purpose has been from the many conversations you and I have had over the years, but hearing it sort of distilled down from you, and frankly, it has to be really fulfilling to see that like that primary purpose of your professional career seems to be really playing out in a big way. And I think in truth, most of the impact is still yet to come. So thank you for everything that you've done. For all of you who want to learn more about Anne's lab and her research, you can visit the Broad Institute at broadinstitute.org. And we will be back soon with more bold ideas and brilliant minds on the next TechBio Talks. See you next time.